Hello everyone, we are going to discuss the next generation science standard HS-LS1-1, which states, construct an explanation based on evidence for how the structure of DNA determines the structure of proteins, which carry out the essential functions of life through a system of specialized cells. This is going to be very fun. Here we go. We are going to focus on the I can statement. I can construct an explanation how genes code for proteins. Alright, let's review from the last video we watched. Remember, we talked about DNA and its structure and its function. So the first question here, what makes up a DNA molecule? Remember, DNA is made up of those repeated nucleotides. It's made up of a sugar, a phosphate, and those nitrogen bases, those A, U, C, G, those T's. Number two, what are the functions of DNA? Remember, DNA is what makes us who we are, gives us our hair color, our eye color, and DNA also codes for proteins that are essential for our body. We're made up of proteins all over throughout our body. Number three, where is DNA found within an organism? DNA, remember, is found within the nucleus of our cell. Alright, before we can talk about how a protein is made, we need to understand what a gene is. A gene is a segment of DNA that helps code for a specific trait. And a gene is found within a chromosome. So here we have a chromosome. If we unwind that chromosome, we have that whole DNA strand. And a segment of that DNA strand is what we call our gene. And what this gene does is it helps make proteins for our body that will code for those traits, that will make those traits, and it needs to use a molecule called RNA to do this. RNA stands for ribonucleic acid, and this is very important in protein synthesis. This structure. It is found within the nucleus of the cell, and it's found floating around in our cytoplasm of our cells. This right here is a picture of RNA. So let's look at how RNA structure is compared to maybe how DNA structure is. So RNA is a single-stranded molecule. So here, it's just a single strand, one strand there. And it's made up of subunits called nucleotides, that name sounds familiar, of nucleotides of that sugar, that base, and that phosphate. Okay, and each nucleotide contains the phosphate, a ribose sugar, and a nitrogen base. Alright, so here we have a picture of DNA, and we have a picture of RNA. So let's note some differences here. RNA is single-stranded. DNA has that double helical shape, so it's double-stranded. Okay? If you look at the nitrogen bases between the two, they both have C, they both have G, they both have A, but they have a different one down here. We have something called uracil in RNA, and then we have that thiamine in DNA. So there's a couple differences between DNA and RNA. Alright, within our cells we have three different types of mRNA, or three different types of RNA that are essential for protein synthesis to occur, for our body to make proteins. The first one's mRNA, stands for messenger RNA. That one plays a very, very vital role, and we'll talk in more detail about each of these. tRNA, transfer RNA, and then rRNA, ribosomal RNA. We will get to those shortly. Alright, so protein synthesis there's two processes within protein synthesis. There's transcription and there's translation. So to kind of go over what these mean. So transcription is taking a strand of DNA, a double strand of DNA, and you're transcribing, or you're making, a copy of an mRNA molecule. So a single strand. Remember, mRNA is single-stranded. So we go from DNA and we make a copy of mRNA. That's the first step in protein synthesis. The second step is translation. In translation, then, we're going to take that mRNA strand and we're going to build proteins. And it's a lot more detailed than that, but that's a brief overview for you for now. So like I said, transcription is the process of taking a DNA strand and making an mRNA strand. This happens within the nucleus of our cell, and it's the very first step in the process of making proteins for our cell. So if we take a look here at this picture kind of does it's a really good description of the process of transcription. So we have a strand of DNA. Here's our strand of DNA. And what happens is we use that DNA strand to make a copy of an mRNA molecule, which is then going to be used to start making that protein. So let's go over the brief overview of what transcription looks like. Alright, so as I said before, transcription, we are taking a DNA strand and we are going to make an mRNA copy from that strand. So here's how it works. We have a DNA strand here. Okay, on our DNA strands, we have something called a promoter region. Okay, what happens is an enzyme in our bodies will attach to that promoter region on our DNA strand, and it's going to signal the start of transcription, the start of making that mRNA copy. And an enzyme called helicase is going to go through and it's going to unzip or break all of those hydrogen bonds that are binding those bases together. Okay, that's going to make it so we can separate that molecule and start creating a new strand of mRNA. So what we have then is here's what it's kind of going to look like. And this helicase molecule would still be going through and breaking those bonds. So we would have something that looks like this. And what happens is we have something called, I keep drawing on that, something called a template strand, and that would be this top strand here that we're going to use to make that mRNA copy. So that bottom strand is really not going to serve a purpose for us here. So to make that copy, we have something called RNA polymerase. Okay, oops, sorry about that. RNA polymerase. And what RNA polymerase is going to do is it's going to take free-floating nitrogen bases that are just floating around in our cell, and it's going to bring those nitrogen bases and match them up with the correct base on the DNA strand. So if we have a C on a DNA strand or a G on a DNA strand, we want a C that's floating around. Okay, if we have a C, we want a G that's floating around. If we have a T on the DNA strand, we want an A that's floating around. And if we have an A on a DNA strand and we're looking to create an mRNA molecule, remember A is going to now match with U. Okay, A matches with U instead of T when we are creating an mRNA strand. Okay, so this process is going to continue down the strand until we reach something called a termination signal. So further along down the strand, we might reach something called a termination signal. What that termination signal is going to do is it's going to tell everything to stop. And that's going to be the end of the process of transcription. Then what's going to happen, just get all this stuff out of the way, then what's going to happen is we are left with this mRNA strand, which is kind of attached loosely to the DNA strand. So this is our template strand here. And here's our mRNA that's beginning to be copied. What's going to happen is that this mRNA is then going to release itself from that DNA, and it's going to take itself from the nucleus where it is right now, and it's going to go to the cytoplasm where it's going to continue to make the proteins in translation. Okay, so to quickly just go over those steps again so you have them in front of you here. Transcription, what happens is an enzyme attaches itself to the promoter region of the gene. Then the DNA is going to unwind with the help of that enzyme called helicase. And one strand of the DNA is now going to serve as the template strand. Okay, RNA polymerase is then going to add free-floating nucleotides to the complementary template DNA strand. And then we have an mRNA strand is transcribed until the termination signal is reached. And the two strands of DNA will rejoin by the help of something called an enzyme called ligase. and the mRNA strand is going to fall off and it's going to head to the cytoplasm where translation is then going to occur. Okay, so if I were to ask you to transcribe the template of the DNA strand below, if you were given this situation, so this is a template strand of DNA and we wanted the mRNA copy of it. So we would go through and we would say A is going to pair with, remember A pairs with U when we're creating an mRNA strand. I'm going to use a different color. T is going to still pair with A, C with G, G, U, A, C, C, U, 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 A, U, C. So if we're creating an RNA strand, we're never going to have a T. Okay, so this slide says proteins. We want proteins, but we first need to understand really what a protein is. Proteins are polymers of amino acids. So if we have a bunch of, say these are little amino acids bound together, that's what makes up a protein. Okay? Between those amino acids, we have something called a peptide bond. It bonds those amino acids together. Okay? We only have 20 different of these amino acids in our cell or in our bodies that are allowed to be made. Okay? But think about this question. There are hundreds of thousands of different proteins within our bodies. How could this be if we only have 20 different amino acids? Well, think about the letters in the alphabet and how those can be arranged in so many different ways to make so many different words. That's just like the amino acids. They can be arranged in many different ways to make many different proteins for our cells. Alright, so now we're to the step of translation. So the goal of translation is to make a protein using an mRNA, which we just created, tRNA, and rRNA. So mRNA will go from the nucleus into the cytoplasm to make proteins. So let's take a close look at this picture here. So in transcription, we took that DNA strand and we made that copy of that RNA strand. Now in translation, we are going to take that RNA and we're going to create proteins with the help of these amino acids here. Okay, so the steps of translation. So the very first step, the mRNA is going to leave the nucleus and it's going to bind to a ribosome. So it's going to look something like this. You're going to have an mRNA strand, okay, with all these letters on it. Okay, and a ribosome is going to look something like this, and that contains that rRNA that we talked about. And that's going to bind to that RNA strand, and it's going to bind to it at something called a start codon. It's going to look for those first three letters, always AUG. It's always going to look for that. Okay, we call those three unpaired nucleotides on that mRNA strand, we call that a codon. So this right here, AUG, is a codon. CUG is another codon. Okay, those three letters are going to code for what we call an amino acid. So for example, AUG is going to code for the amino acid methionine. So if you look at your codon wheel that your teacher provided for you, and you look at the letters there, you start in the very center. Okay, the first letter you look for is A. Okay, A would be one of those four in the center. Then you're going to go outward from A and you're going to look for U. Then you're going to stay in that little narrow area and you're going to look for G. And that's going to point to the amino acid methionine. So if you're going to look for the amino acid for the next one, for CUG, take a look at your codon wheel. If you look at C and then you narrow down to U and G, you're going to code for something called leucine. Okay, that's a codon, and that's how you code for amino acids. So the very first thing, we attach that rRNA to the start codon, and then we move on from there. Okay, so let's look at more steps of translation. So we still have, I'm just going to make this a little bit smaller and make our molecule bigger here. So here we still have the ribosome attached with that rRNA. Okay, so what happens then is an amino acid is carried by a tRNA molecule. This is going to be our tRNA molecule, this structure right here. Okay, So this amino acid, or this tRNA molecule, is carrying this amino acid, methionine, and that's what MET is standing for, over to the mRNA strand that we have here. So what happens is this tRNA carries an anticodon, okay, which I haven't drawn yet, an anticodon that is complementary to this codon here on mRNA. Okay, so an anticodon is a group of 3 unpaired nucleotides on a tRNA molecule. So if it's complementary to this mRNA strand here, we are going to have UAC. Okay, and once those two link together, once they kind of link together, that's what's going to cause that tRNA to know to bring and attach that methionine to that position, that first amino acid. All right, so the anticodon on the tRNA finds the correct codon on the mRNA. So, for example, the anticodon to AUG's codon matched, and then that signaled that amino acid methionine to be carried there. Next, the ribosome, as you can see, has moved down the mRNA strand to the next set of codons. Okay, the tRNA is going to do the same thing. It's going to bring its anticodon, and it's an amino acid here, leucine. If you look on your list, you can see that CUG codes for leucine. It's going to bring that over, match them up, and you can see that now we have, I'm going to move this up a little bit, we have a chain of amino acids forming. Okay, this is going to continue. This is going to continue and down, it's going to continue down the DNA strand until we reach something called a stop codon. Okay, once we reach a stop codon, that's going to signal that the protein is done forming. Okay, one thing I didn't hit on and I want to quick mention here is we have a little bond that forms between those amino acids. That's what we call the peptide bond that we were referring to earlier. Okay, so we continue down, the protein continues down the strand. We finally reach something called UAA, which is a stop codon. If you look on your codon wheel, that codes for stop. So what's going to happen is that leucine and that methionine are going to break free. Okay, they're going to break free, and that's what's going to form a protein. Obviously, it would be a much longer chain of amino acids than just those two, but to just kind of show you the brief overview, that's kind of what I did there. Okay, so now we've created a protein from that strand of mRNA. That is translation. Okay, so now here I have a little practice problem for you to do. So it says transcribe the template DNA strand. So we're going to transcribe that template strand here. And then it says now after that you're going to translate the mRNA strand that you create into a protein. So I want you to pause the video and then check and see if you got the correct answer. Okay, go and check your work and make sure that it is right and matches my work here. And you might need to pause the video because I'm going to continue on. So let's be reminded of the importance of proteins within our cells. So for example, here is a red blood cell. Red blood cells contain the protein hemoglobin. The protein hemoglobin carries oxygen throughout our body so all of our cells can obtain the oxygen they need. Okay, this one right here is a zipper. What I'm trying to get to there is that enzyme helicase we talked about that helps in protein synthesis. It's what unzips the DNA strand so we can make that mRNA. So really, a protein helps make a protein. Okay, this picture here, I have a doorway. What I'm thinking about there is in our cells, we have little protein channels made of something called transport proteins that are like a doorway. They help things enter and leave the cell. Okay, cells don't always need the same amount of things on the inside as on the outside, so things are transporting back and forth constantly through these transport proteins. Then I have a picture here of the digestive system. Remember earlier we talked about the protein pepsin found within our stomach that helps break down food particles that we eat so we can obtain those nutrients. Also, proteins are the building blocks of who we are. You wouldn't have a certain skin color because remember, melanin is a protein that makes your skin a certain color. Okay, proteins are what make your eye a certain color, your hair a certain color. Okay, so they are essential. They are the building blocks of living things. So to conclude, remember, transcription is the process of taking a DNA strand and making an mRNA copy. In translation, then we take that mRNA copy and we end with proteins. All right, and here are the credits, and I hope that was helpful for you all, and have a very good day.